colleagues, friends, and most importantly, David's friends, family and supporters. I'm Ian Martin, I'm the DVC academic, and it's my very great pleasure to welcome you here to tonight's inaugural lecture. Before I get on to introducing David, I'll just put some context around inaugural lectures. So, the inaugural lecture has a very long and mostly distinguished history. It was part of the tradition of medieval guilds when an apprentice became a master, and it moved from being part of guilds to universities when the first medieval universities were established. Tonight is a one-off event. We're here to celebrate David's achievements and hear a little bit about his academic story and family story to date. But it hasn't always been quite like that. There were certain expectations around inaugural lectures where the lecture didn't feature very strongly and the eating and drinking surrounding it featured considerably more strongly. But there was also a tradition that I do wonder whether we should resume, which is the person giving the inaugural lecture actually inherited a few university responsibilities for a fixed period of time. So, at least in one university, the person who gave the inaugural lecture had to preside over university disputations for 40 days and 40 nights. So, David, you've got an interesting month and a half coming up. The other thing that was expected was certain gifts to university administrators. A tradition I definitely think, and looking at my colleague Les Field, DVCR, who are both nodding that we should resume. And the other thing that was expected, that you had to entertain the university for food and drink for a certain number of days as well. With a university of 6,000 staff and 52,000 students, that might be somewhat more tricky. But the common feature in all of those traditions was that this was to mark an important rite of passage for someone in their career, their professional career, their academic career. And being promoted to our highest rank, that of professor, is undoubtedly an important rite of passage to celebrate. And we're here today to celebrate that for David. So, David's a researcher at the interface between statistics and ecology, with a passion for improving the analysis methods used to study the natural world. He's Sydney born and bred, currently lives with his family in a flat in Epping, just 1 kilometre from where he was born. He studied at the University of Sydney, at one point working as a party clown on weekends, and in between that completed a double major between pure maths and biology with honours, and in brackets, but certainly not in brackets in terms of an achievement, the University Medal in Plant Ecology. He saw the light and moved to stats soon afterwards with a Master of Arts, Mathematical Sciences and a PhD at Macquarie University, and hence the robes. And that was shared between ecology and statistics. He came to this great institution as a lecturer in 2003, just weeks after submitting his PhD thesis and getting married. 2 events separated by just a couple of days, which for anybody is a pretty prodigious achievement. And he's been a key part of our academic life here since then. He's made huge contributions to the study of size variation, ecological communities and their climatic response, and the spatial distribution of species. His contributions have had substantial influence on statistical practice across disciplines, used in over 3,000 ISI articles in statistics, ecology and other areas of biology, earth sciences, agriculture, medicine, chemistry, psychology, engineering and physics. Last year, he was awarded the Christopher Hyde Medal for contribution to statistics and a Young Investigator Award from the American Statistical Association. He's received over $3 million in ARC funding since 2007. He's given over 40 invited talks and has just started a 12-month stretch where he will be keynote speaker 3 times across 3 countries. As well as being a fantastic academic, he's taken social media and the internet as a tool to extend the reach of his work, and he's written provocative pieces for ecologists, including a YouTube video comparing multivariate analysis to Rick Astley. A quite frightening comparison that I really hope is not repeated tonight, although I really do fear, and it gets worse. and a blog post describing Maxent as looking a bit like Beyonce, but really just Janet Jackson in disguise. Again, euphemisms that really probably only could come from a very special academic. Ladies and gentlemen, I now invite David to give his inaugural lecture. David.
Thank you very much, Ian, and thanks everyone for coming. And I also should thank the VCs unit that organised this event. It's much appreciated. And I guess while I'm thanking people, I guess I've got a slide of thanks. I'm going to start with my mum. Mothers tend to do a lot for you over the years, but the most sort of influential careers discussion I had was with my mum. She doesn't remember it at all. But I was thinking of doing thinking of a career in science, maybe something kind of a bit biological, and I was worried about I wasn't sure about job prospects. I mean, at that time, I didn't realise how, with a science major, there's actually quite good employment prospects. If you've got like a major in maths or stats, then, you're beating them off with a stick sort of thing. But at that time, I didn't know that. And she told me to do what I enjoyed. If I enjoyed it, I was probably going to be good at it, and then I'd probably get a job out of that, hopefully. Well, she sort of gave me a big vote of confidence, and then I guess here I am. I'd also like to thank my family. I've got 2 little ones, and my wife Lorena does sort of most of the hard yards, and so I'd like to thank her. And I guess my parents also help out a fair bit, and different times. And I guess my friends and family more broadly. I think I'm quite lucky to have to be surrounded by a bunch of good people. And maths and stats it's like a really nice place to work. They're good researchers, but they're good people to work with as well. I don't have a picture of them, so this. You Google School of Maths and Stats, and this is the best you can do in terms of getting a picture of your colleagues. They're all sort of around here proving theorems and finding counterexamples. It's a pleasure to work with them in a really sort of stimulating work environment. I'd like to thank my collaborators, my students and postdocs, who are a lot of fun to work with. They're kind of like my work family, and I get to be like the work dad, or the work sort of cranky grandpa, depending sort of how things are going. But they're a lot of fun to work with, and they're a really talented group of people that really sort of we get so much more done than I could do, and it ends up being better quality than what I could do as well. But somehow I get to take the credit for it. But they're a pleasure to work with. And who else? We've got the Australian Research Council. So there's a lot of really good researchers out there that don't get ARC funding, just because there's not enough to go around. And so, I'm very fortunate that for whatever reason, ARC sort of chose to back me, because that's really important in anyone's career when that happens. And so, I guess thanks to them, and thanks to my supervisors and mentors over the years. So, they are more or less in chronological order. I don't think any of them is really to blame for the way I turned out, but they certainly have sort of influenced the way I think about things and smoothed out a lot of the rough edges. So that's it for the thanks. Now it's on with the talk. So I wanted to start by telling you all what an exciting time it is to be a statistician. There are a few jokes in here, but that's not a joke. So when I started high school, this is what computers looked like. It's an Apple IIe, 64 kilobytes of RAM, 64 kilobytes. This digital image is over 100 kilobytes, so this poor computer doesn't have enough memory to look at a picture of itself. And if you go to a high school these days, computers are thousands of times faster, and you can get a million times more memory. So it's been a real game changer in a lot of areas, but certainly in data analysis. So this doesn't seem to be working. It is? Oh, good. Okay, cool. Yeah, so I guess two ways that technology has changed statistics. One of them is in presenting new types of data. New types of data give us new problems for which we need new solutions. So if you've come across false discovery rates, for example, this was a technique that came out of thinking about the problem of how to identify which genes are causing cancer out of sort of thousands of data for thousands of genes. And it's a technique which is now used across the sciences. So that's kind of one thing. New technology gives us new data, new problems, new solutions. But new technology also makes it possible to do all sorts of things that we just couldn't have dreamt of sort of 10 or 20 years ago. New technology both in terms of new mathematics, new computing techniques too. So it's just been a real game changer for the discipline. It's a really sort of exciting time to be a statistician, which of rewriting the rule book in many ways we have been over the last decade or so. And there's also so many cool things that you can do with stats. So here's just a few of them, a few things that I've been involved in. So one of them is modelling where species are as a function of climate. So then you can look at the question of where the species, what's going to happen to the species as the climate changes. So with this particular species, like most of them, under warming scenarios, if it wants to stay in the same sort of climate that's living in now, it's going to have to move southwards. So I did some consulting for New South Wales Police. They were interested in factors that are associated with whether or not someone's going to confess to a crime. I can't tell you the results, it's classified information. So there was someone with an idea for a new casino game, and they needed to know the expected rate of return so that they could work out whether it was the casino was going to get as much money as they wanted to out of it. I've talked to Tracy Rogers about how to analyse whale call data. I've done some work on megafauna, so sort of the bones, what you can find out about the way animals, extinct animals lived from looking at the fossils of their hands. Dinosaurs, all sorts of different things. And so they're quite interesting and quite different sort of applications you can be involved in as a statistician. But also the methodology is quite different for each of these different problems. So it's quite intellectually challenging as well as the end applications being interesting. So it really is an exciting thing to do. John Tukey had a way with words. He said that the best thing about being a statistician is that you get to play in everyone's backyard. This is the guy that also coined the term software back in the sort of the 50s, early 60s. Hal Varian, Chief Economic Officer at Google, became an instant celebrity amongst statisticians when he said that statistics was going to be the big, sexy job of the 2010s. I guess his thinking was that a good job, a big, sexy job, is one where there's lots of demand for what you do, but there's not that much supply. And everyone's got data, but not that many people know what to do with it. Statistical thinking will one day be as necessary for efficient citizenship as the ability to read and write. Well, stick to sci-fi, buddy, because it's not the way it's worked out. So let's talk about statistical literacy. I've got a pop quiz for you. So, should either of these guys here be worried about the results of a poll of about 1,600 people, where the Liberal National Party vote was down 1%, shortened dissatisfaction rate was up 3%? Yes, no, maybe? Yes? Anyone for yes? Anyone for no? Anyone for maybe? Yeah, I understand it's hard to take the square root of quadratic functions in your head. But, well, not really. I guess anytime you take a sample, you don't have the whole population, you've just got a sample. So you're going to get different answers for different samples. And you can use probability to estimate how much variation there is from one sample to the next. And in this case, it's going to be the amount of uncertainty we have. We could put a number on it and say we're pretty sure we've got the right answer to within about 3.5%. But that's 1% plus or minus 3.5%, or 3% plus or minus 3.5%. So we don't really know that this headline is true. If you asked what I really thought, or if you looked at a few different polls, I guess there probably was some evidence. This is from a few weeks ago when the Royal Commission stuff was on. And so there probably was a bit of a shift in shortened dissatisfaction. Other polls sort of seem to have said the same sort of thing. This one here was probably random noise. And that's probably something that you could say about half the poll headlines you see in the paper. I mean, it's hard to know exactly, but a lot of it is just sort of random noise because different samples give you different answers. So you get something that looks like a change, but probably wasn't. Okay, here's another one for you. Is this graph evidence of a Sydney housing bubble? So the bubble argument is that if you fit a straight line to data over the last 30 years, we're above the line. So how's there overpriced? Yes? No? Scared? All right. So yeah, this one's a no, and certainly not from these data. I guess so. Housing price, profit, size, abundance, these are all sort of things that make sense as multiplicative processes. So it makes sense to think of them as an outcome of a bunch of things we're multiplying together. You talk about rates. You talk about a 5% increase in price. You don't talk about a $100,000 increase. It's all rates. And so it makes sense when you're talking about something which is an outcome of a set of things being multiplied together. They make more sense on a logarithmic scale. If you put things on a log scale, then a 10% change is the same distance everywhere along the scale. So if you look at house prices on a log scale, suddenly we're a bit, it looks like they're sort of, compared to historic value, historic rates of increase are actually below trend at the moment, which is not sort of the story you're hearing in the papers. I'm not an economist, so I'm sure there's other arguments to it about affordability and prices elsewhere, but certainly comparing the price now to historic data, you wouldn't say we're in a bubble. Okay, so there's 10 bosses at your work. They're all male, and let's say they're drawn from a population of 50-50 males, females. Recall that if you take a half, raise it to the power of 10, it's 1 on 1,024, about 0.1%. So we're 99.9% sure that there's gender bias going on here? Or would you say another way of saying it, are we 0.1% sure that there's no gender bias? The answer to this one's no, because that probability was the probability of getting 10 male bosses out of 10 if there was no gender bias. That's not the same thing as if you reversed what event was being conditioned on. It's not the same thing as the probability of no gender bias if you got 10 male bosses. I mean, say you meet an Englishman in the street, the chance that he's the VC at UNSW is small. But if you meet the VC in the street, chance that he's an Englishman is pretty large. So reversing the order of conditioning, it gives you a completely different event. So that they aren't the same thing, and we can't just say that we got 0.1% chance of no gender bias. So these are just a few examples of things which you see around the place a lot in the media and in science. Estimates from samples come with error, and we often sort of forget about that and just talk about the trend without thinking about uncertainty. Multiplicative process, they most naturally thought of on a log scale. Alan Kohler, I'm a big fan. He almost makes finance interesting, but he's always taking these multiplicative processes, putting them on an arithmetic scale, and drawing straight lines on them. Yeah. Find that a bit annoying. So the order of conditioning matters in probability, and I guess a place you see scientists sort of get this wrong a lot is when talking about p-values. A large p-value doesn't mean that you've got a high probability that the null hypothesis is true. When you say that, you're making this mistake of reversing the order of conditioning. So some scientists from CSIRO. It's a bit harsh. No, they wrote a really nice piece for The Conversation, but the title was wrong. So they had evidence. They'd done some work which showed that they had evidence that human activity was contributing to climate change, but they didn't have evidence to make this claim that they're 99.99% certain. They just reversed the order of condition. That's what they got wrong there. So I guess so in my research, I guess there's these two ideas going on. There's this, it's an exciting time to be doing stats. And so a lot of what we do is harnessing new technology to answer sort of important ecological questions. But then on the other hand, it's also tough times in the sense that there are issues with statistical literacy and you know, an ecologist isn't trained like to a high level in maths, and you know, so how are they going to keep up with all this rapid advance in methods? And so this is something that I also try to address in my work, trying to bridge the knowledge gap between ecology and statistics. So an example of this is multivariate analysis of abundances, and that's where the title, the retro night thing, is coming from. So an example here, there were some collaborators went and sampled all over southeastern Australia in grasslands. They were looking at invertebrates, and they wanted to say something collectively about invertebrate communities. What they were interested in was how these invertebrate communities change as the climate changes. So they ended up with a matrix of data. The different places they sampled are in the rows. The different things they sampled are being counted in the columns, different species of bug they were sampling. And they'd like to know across the whole community, you know, how are these communities of invertebrates responding to climate? So this type of data, we could call it multivariate abundance data. It's got two key properties. There's an abundance property. The abundance property is that, I guess the key part of being in abundance is that different species are going to have different abundances. Some of them have a small mean, some of them have small counts, some of them have big counts. And that gives you what's known as a mean variance relationship. If your average is small, your variance is small. If your average is large, your variance is large. And so this is something that you'll always see when you're looking at this type of data, abundances. Another property, we could call it the multivariate property. We've got lots of species and they're correlated with each other. So species interact with each other. So we can't say these things are independent. So we've got a multivariate problem. To make it a bit harder, there's lots of species compared to the number of sites usually. So we could call that high dimensional. That's a term that's often used for this stuff. So we've got high dimensional data that's got this mean variance relationship. So they're the sort of the two things that we've got to try and deal with this type of data. Of these two problems, this one's the hard one. If you went to a statistician in the 80s with this type of data, they would have probably told you to go away and come back with less response variables. So ecologists went away and made up their own methods. So it was quite a clever idea, I think. I think this was a really nice thing to do, especially when you consider the computational constraints that people were working under at the time. So what they'd do is they'd sort of take all these response variables and they'd collapse them down into a single metric that compares different rows with each other. And then they've got a vector of a single variable to analyse, and they could just use standard univariate type ideas to analyse it. So it was a big hit, and it's a big hit today. People have been developing extensions of these methods continuously since then, and it's like if you talk to statisticians about multivariate analysis, you get a quite different story to what ecologists talk about as multivariate analysis, because pretty much everything the ecologists are doing is using this idea. It's textbook stuff. It's taught in undergraduate programs in ecology, this way of doing multivariate analysis. But to put that into context, here are some other things which seemed like really cool ideas in the 80s. Big hair, mullets, Cabbage Patch Dolls, Dave Dobbin, Rick Astley, Colette. So, do you remember Colette? That's a no. I have my ghetto blaster. I think I got a cassette tape here somewhere. This. So that's Colette for you. Do you like my fluoro socks? Got my fluoro socks on. Fluoro was huge in the 80s. We got fluoro socks, fluoro jackets, fluoro beanies, fluoro shoelaces, fluoro fingerless gloves. And this idea of taking a whole bunch of response variables from different distributions with different variances, and applying some metric to collapse it down into a single metric that we're going to use for analysis, I'd put this in the same category as fluoro fingerless gloves. I think it was a cool idea in the 80s, but these days it's just not cool. And I've got a few reasons why I think it's just not cool. One is that it's got poor power properties. So I've got a simulation here where I had lots of species. We've got lots of species, and there's a big effect in just one species, like a 10-fold change in mean for one species, and nothing's happening in any of the others. And we're going to see what happens as we change which of the species has the effect in it. What we'd like to see is high power. Doesn't matter what species we've got. What we actually get with these methods is no power nearly all the time, and then there's a few species that have high variance where we do have good power. And the reason for that is that these methods aren't dealing with the mean variance relationship. So remember we had this big mean variance relationship. They're not dealing with it, and so they're being dominated by the high variance species. You could try transforming your data to deal with this problem. It can help, but it's not going to deal with it because the thing about having counts with lots of zeros in it, you just can't get rid of this mean variance relationship. You can try and make it smaller, but you can't kill it. So you've always got this issue. Okay, next problem. We can't detect special mean structures. So there's some special mean structures that ecologists care about. One of them example is composition, if we're interested in changes in composition. So by composition we mean changes in relative abundance. Say we're interested in testing, are there any changes in relative abundance? Mathematically, it looks a bit like this. We're saying is our mean vector some scalar multiple of some common mean vector. We're going to do a simulation where we've got a twofold change in total abundance, but no change in relative abundance. And we're going to see how well the methods people are using and have been using for decades, how good they are at testing at recognising when there's no change in community composition. These methods are commonly referred to as community composition analysis, so you'd think they'd be good at it. You want to be in the grey zone. The grey zone is where you want to be in this case. This is a type 1 error simulation. Almost nothing is in the grey zone. So there's one here which is doing okay. That had poor power. That was one of the methods before that had this weird power property going on. This one's doing okay, but the other ones, the methods which are really commonly used, like the software default sort of methods, they're not even close. Not cool. Okay, so I guess another thing, say you give two people the same data set, the same research question, sometimes they'll give you completely different answers, and they'll be able to justify those answers by using sort of the previous literature saying this person says, use the Alt-Gower metric or fourth root Brake-Curtis, and they're getting completely different answers. So, for example, some coral abundance data, and we've got an ordination here. We're looking at what's happened in 1983 compared to 1981. There was an El Nino year in there, some coral bleaching between these two sampling times. And over here you'd say, well, the disturbance from the coral bleaching has increased the variance, it's increased dispersion. Over here you'd say the opposite. So that's not cool. All right, so what's missing? What's missing from this picture? All we need is a model. All we need to make this thing work is a model. So it's and this idea of as a first step, if as a first step you're collapsing all your variables down into a single metric, that prevents specification of a model for the underlying data generating mechanism. We want to model the process back here. We want to start talking models there. We don't want to do this and then try and start picking up the pieces later. So there's something just philosophically wrong with this idea of as a first step, reducing your data using distance metric. Of course, back in the 80s, we couldn't do this, but there's been lots of things that have happened since the 80s. Good and bad, but here's some of my highlights. The internet, smartphones, Shane Warne, and Katy Perry's Shark Dancers. In terms of statistics, I guess Markov chain Monte Carlo is going to be pretty much top of everyone's list as a big advance in statistics. And that really sort of revolutionised Bayesian stats. There's modern methods of numerical integration, all sorts of different things. I'm partial to this one. I must be a 90s man. But you know, there's all sorts of things that make it a lot easier to do maximum likelihood, so non-Bayesian methods of analysis. This is a funny one to include on your list, but I quite like this idea of using probability integral transform residuals. So if you've got count data and you want to do a residual plot, if you just do the naive sort of count minus fitted value divided by standard error, you get something that looks like this. You get these weird lines. There's a weird line here corresponding to all your 0 counts, a weird line here for all the 1s, and a line here for the 2s. And when you look at a residual plot, we want to be looking for no pattern, but we've got a pattern just through the discreteness in our data. So there's got to be something better we can do, and in the end, the thing we can do is called a probability integral transform residual. So probability integral transform, this tells you the probability of getting less than or equal to a particular value. So here we got about a 0.7. Chance of getting less than it is about 50%. Chance of getting less than this one's about 10%. Chance of getting less than this one's sort of almost 80%. So the neat thing about this is that if your probability model is right, then it doesn't matter what your distribution is along here. The values up this axis are standard uniform. They're evenly distributed between 0 and 1, equally likely to get any value between 0 and 1, if your probability model's right. We'd like to be able to do that. So that'd be a good thing to use for residuals, because it doesn't matter what distribution our data has, we can put it onto a common scale using this transformation. But with discrete data, you're only getting, like, if you've got counts, it's a 0 or a 1 or a 2 or a 3. So you're only ever going to get this value or this value or this value. It's jumping around. It's not uniformly distributed up this axis. So a neat trick we can do is to just add some randomness. So instead of just mapping across up here, we can generate a value at random between here and the previous value for the cumulative probability. And if you do that, then if your probability model is right, things are standard uniform up this axis. You're equally likely to get any value between 0 and 1. So that's pretty cool. When I first found out about this idea, I thought it was awesome. It's an idea that actually sort of was originally sort of, I first saw it in the 80s, like in a paper from the 80s, but the idea of applying it as a method of doing residual plots. There was a couple of Queenslanders in the mid-90s. So instead of getting a weird plot like this with your lines here, you suddenly get something like this, if your model is correct. So you can construct these plots and look for a pattern, and if you don't have a pattern, that suggests that you're on the right track with your model. So I think they're awesome. What else is there? There's heaps of other things that have happened since the 80s. Here's just a list of a few things which are going to come in handy when it comes to analysing this type of data that we've got, these multivariate abundances. So if you're building a model like this guy, then there's, I guess, two main things that you're going to need to think about. What's the research question? Because, yeah, it doesn't, the model that you use is going to depend on what the purpose is, like what you're doing it for. And then there's also the data properties that we've talked about. We also need to think about that when it comes to building a model. So here's a few different purposes here that we could be building a model for. And my group has been working on each of these proposing models It's showing how they work as compared to all competitors sort of over a period of several years now. And we've pretty much ticked all these boxes. I'm going to start with this one. I'm not going to go through all of them, we don't have time. Well, I could if you want to. Say you want to test some hypothesis, you're testing for environmental impact, or I guess for this data set, we'd be looking at is there an effect of transplanting, because we'd like to transplant plants from one site to another to try and separate the effects of climate from the effects of host. So we've got a hypothesis to test. What are we going to do? To handle the abundance property, it's pretty easy to do. You use what's called a generalized linear model. Generalized linear model, if you haven't seen it before, but if you've seen linear regression, it's pretty much just like linear regression, and then instead of assuming equal variance, you add a mean variance relationship to it. You say, my variance is increasing with my mean, it's like this function here, so I'm going to assume that, and away I go. So a GLM, you've got two equations in there. There's your linear regression type equation there. We've got a transformation of the means, but it's just like a linear function of x. We've got a mean variance relationship here, and we're off and racing. So that's how we handle the abundance property. How are we going to handle the multivariate property when it comes to testing? Well, I guess that's a tricky one, and our first shot at this problem was to use resampling, where you resample rows of your data. So in order to test a hypothesis, you need to generate a null distribution for your statistic, and we're going to do that by simulation. We're just going to simulate data to match the properties of our observed data. There is the question about how we're going to do that when you're looking at GLMs, and we proposed a new method of residual resampling that will work for GLMs. And this one works using the pit residuals that we talked about before. So you've got observations that could become from different distributions, hence they've got different step functions here. But by computing the pit residuals, if our model's right, then these things all have the same distribution of this axis. So we can resample any of those with replacement and map back down to get a resampled value, which should then have the same distribution as the data we originally sampled from. By keeping them together by resampling rows, by using this approach, by resampling rows, we account for the correlation. So this is a way we're going to handle the correlation in our data. So that's one thing we can do. Ordination. So that's another thing people often like to do. It's pretty much dots on a page. It's a way to visualize your data, but it's a nice way to try and see the signal from the noise, because if you look at each of your variables separately, they're bouncing around all over the place. There's a lot of noise in there, and this is a way to try and get at what the underlying signal is. But we saw before that we had these two completely different ordination plots, so we didn't really know what was going on. The reason we had a problem was that there's this mean variance relationship going on, but as well as that, we've got a huge shift in the means. So there was a coral bleaching event where most of the coral died. So in 81, the means are all up here, but then they all shifted down here. And the previous methods, like of doing ordination, they were giving quite different answers because there's quite different variances here. These ones are like, hundreds of times larger variances than these ones here. And depending on how you handle that, you'll get different answers. But a good way to handle it would be to model this, wouldn't it? You just sort of say, okay, I've got a mean variance relationship. I'm going to assume my variance is this function of my mean. I've got some predictors in there, I'll throw them in too. And the way to make this work in the context of ordination is to add these things called latent variables. So latent variables, these are like unmeasured variables. It seems like it's a bit mysterious, this idea that you can throw in unmeasured variables as predictors in a model. Like, how are you going to estimate these things that haven't been measured in the first place? But you can actually do it based on the correlations, because if you've got multiple response variables, multiple species all responding to the same thing that you haven't measured, then you can use the correlations to work out the values. So that's kind of the idea here. We've got these latent variables in there, and they've got two roles in our model. They work as axes for ordination, and they work as a way to model correlation as well. So we're ticking all our boxes here. We've got something that's handling the multivariate property and something that's giving us an ordination. But instead of giving us a plot like this, what's it going to do? Is it going to give us sort of higher variance for 83 or for 81? Gives us about the same variance for both of them. But we've got a big shift in the mean. And that makes sense if you think about the mean variance relationship. It didn't seem to be changing at all when you went from 81 to 83. The thing that was changing was the mean. So we've just got a shift. We've got a location shift, but we don't have a change in dispersion, as far as we can tell. So that's something else we can do, latent variable models for ordination. So how about community composition? So this problem of looking at relative abundances, how do we do that? So is there a change in relative abundance? Instead of just looking at total abundance, does relative abundance change? How are we going to look at that problem? Well, that's really easy, actually. If you think that the total abundance might be changing in different places, you just put a row effect in your model. You just say, look, I've got a row effect that says that there's lots of counts there and not many counts there, and you're done. So we take our GLM, we add a row effect, saying that when we sample in different places, we might get different values. And then everything else in the model is looking at relative abundance. So this I think could be quite useful, for example, in situations where you don't have control over the amount that you sample. So sometimes, like in marine grab samples, you're just sort of sampling off the bottom of a lake, and sometimes you get a good one, sometimes you don't get a good one. And so what we really want to do is look at relative abundance. You want to analyse the relative abundance, and you want to have a term in your model to take into account the fact that just sometimes you get a good one, sometimes you don't. So that's a really easy thing to do, just adding this term. It's not that hard once you've got a model, and it makes all the difference. So instead of all these curves going all over the place, we're staying pretty much where we want it to be when we use a model as a basis for testing for composition. Going back to the coral data, so I guess this time we're going to look at the mean abundance for each species. You can see that most of the species have a big drop in mean between 81 and 83. And so I guess an obvious, these ones here, they've just gone altogether. But most of them have a big drop. And so I guess a natural question to ask for this data set would be, well, was there a change in composition, or was it all just everything just getting smashed? And that's an easy question to answer using models. We just put a row effect in there and see what happens. So whereas we had this big shift between the means there, once we put a row effect in there to account for changes in total abundance, there's not a whole lot going on. So it's clear that for this data set, the main story was just that all the coral died. There wasn't an increase in variance or anything like that. It still just got done by bleaching. All right, there's heaps of other things you can do with models. So you could use species, you could put traits, predictors on species. You could put predictors on species to try and explain why you get differences for different species in different places. So annual precipitation is important for the bugs, and as it turns out, the ones with big heads like it more when there's less rainfall. So this is something that you can do with models that we couldn't do otherwise, because with a distance-based approach, as a first step, we'd collapsed everything together. We'd lost the species information. You can classify species, classify sites using finite mixture models. We can look at correlation networks. This graph was drawn when my daughter was on my lap, hence the pink. So this one is about looking at which species are related to which, just using the data on all the different species, like which species occurred together. We can use that to draw a graph of the network for which species relate to which. It's hard to do for abundances, like you can't just pull stuff off the shelf, but Gordana Popovic has been she's doing a PhD with me, and she's been working way through that one, making some great progress. So we're at a stage now, like with model-based approaches, where we can do most of the things that people want to do, And there's a lot of new things that you can do with models that you just couldn't do using the older distance-based approaches. So there's methods around for all of these different applications and more. And there's not just methods, there's also sort of free software. So this isn't something where it's like on the horizon, something to look out for in a few years. You could download some software and just do some analyses with this stuff on the way home on the bus if you'd like to. It's your call. So that's a little bit about models. And so what do I see is happening next? What's the sort of the 2020 sort of plan, as far as I can tell? Well, I guess looking to the future, obviously we got TV goggles, weed-shaped buildings, touch screens. In terms of statistics and multivariate analysis, I think we are going to see a lot less of this and a lot more of this. And it seems to be happening quite quickly. Just in the last sort of 5 years, there's been some huge changes in methods that are available and in practice, in what ecologists are actually doing. And I guess if there's anyone here who is teaching this stuff in second year to ecologists, like these distance-based approaches, I mean, there is a chance that by the time these students come out, graduate with PhDs, those methods will be redundant. So it is happening quite quickly. Yeah, so that's not to say that everything's done. I guess a few things that I'm currently working on, I'm thinking about testing methods when you've got lots of parameters in your model. It's not an easy problem. And there was a linkage grant that was recently funded to look at hierarchical methods for classification. So there's a couple of things. And I guess spreading the word is something else that's it's something that I think a lot about, how to help sort of bring ecologists sort of up to speed on sort of where stats is at in terms of multivariate analysis. One way to do it is through software and documentation. Another way is through the internet. So, I mean, just as technology's revolutionised science, it's revolutionised the way we do science communication. So I made a video, it took me a couple of days to do it, and it's been seen by more people than I would have talked to about my research face-to-face in my career to date. So, you know, it's a really powerful medium, the internet, and if you've got a message to get out there, it's worth giving it a shot, for sure. So, yeah, I've got a textbook plan. I remember saying to write in 2015 last year, but it's gonna happen. And I guess workshops for ecologists, like we've got sort of a couple of things that we've been developing and that we can, to help sort of train ecologists in sort of modern methods of doing things. And I guess I've also organised a couple of conferences. There was one a couple of years ago, and there's a follow-up in December this year. And really the idea of it is just to get ecologists and statisticians in the same place, looking at the same sort of problems to try and help sort of bring people a bit closer together in terms of the way they think about things. That's it.
Thank you very much, David. So my name's Merlin Crossley. I'm the Dean of the Faculty of Science here, and we've just had a fabulous lecture by you. I'm enormously grateful. You started your talk with thanks, and I want to thank you for all the things you've taught us. You've taught us a lot about understanding political polls. You've told us about the police. You didn't tell us, but I think there's probably usually not much of a correlation between the confession and whether the person actually committed the crime. I'm not sure. You've told us about Alan Kohler, who I think is a great television hero, but his graphs will never be the same to me. You've told me about Katy Perry's Shark Dance, which you didn't actually do, I understand. And I thank you for all that. Most of all, I want to thank you for not playing Rick Astley, which is fantastic. David kept mentioning the period of the 80s when he grew up. To me, he's one of our youngest professors, and I think you must have been very young in the 80s at a time before your musical tastes matured. So I think but there's still great hope for you. I'm thinking the secret, it's interesting, you said modestly the ARC funded your work for whatever reason. I think we should make no mistake. They fund David's work because of its excellence. I think its excellence and the way he is so contemporary, using new emerging technologies to tackle problems in a new way. I think there's a perfect time for each person. And I think you, David, if you gave away the Ghetto Blaster and got an iPod, just imagine how your musical and dancing could progress as well. I think one would ask, what is the probability that we have professors like David here doing this sort of work? It's one of those things where it's not a fair question, because we don't choose our professors at random. We choose them by the quality of their work. And I'm very proud at this university, we have extraordinarily good environmental science and ecology, and we have extraordinarily good mathematics. And my job is meant to be to bring people together so that they can work together to discover things about coral bleaching, about wide-headed insects doing well after rain, about changes in climate, about all of these complexity of things in people's backyards. But it's made easy when the people do it themselves. And I'm hugely grateful that you've done that, David, and I'm hugely impressed by your achievements. To make no mistake, I've watched David go through the various promotions rounds, and every now and then he says, what do I need to be promoted? And I say, all you need to do is get an ARC grant in statistics, and he ticks it off. Get an ARC grant in ecology, he ticks it off. And if you could win the Christopher Hyde Medal from the Academy, that would also help too. And he ticks it off immediately. I'm kicking myself I didn't ask him to win a Fields Medal, because I hope he'll be working on ticking that off in the future as well. So to close, I just thank you all for coming, friends, colleagues, the families who've come, and the dignitaries of the university. We've got 2 Deputy Vice-Chancellors, Ian and Les. Thanks for coming as well, and the various staff and friends, because it was a terrific occasion. Thank you very much, David.